Okay, the next topic is permeability methods. In this permeability, again, it is divided into, or in this we will discuss different in vitro methods, in vivo methods, and in situ methods. What is in vitro, what is in vivo, what is in situ. So I will discuss all the methods in this class. So first I will explain what is absorption. What is absorption? Absorption means it is a process by which one thing is absorbed. Means it is a condition in which something takes in another substance. Okay. It is a condition in which something takes in another substance. It is a physical form or a chemical phenomenon process, or means in the physical or chemical phenomenon, or it is a process in which atoms or molecules or ions entering into the inner part of the materials. Okay. For example, we can say gulab jam. Absorption takes place. Okay. In general example, I'm saying. So coming to the definition, it is a process of, it's a our term, it is a process of movement of unchanged drug from site of administration to the systemic circulation. Means it is unchanged drug from site of administration to the systemic circulation. This is about absorption. It is a process by which one thing is absorbed. It is unchanged form. Next, what is permeability? Permeability means, for example, in our body, the molecules will pass through the biological membrane. Okay, the molecules will pass through biological membrane that will entering into the systemic circulation. Means the molecules transferred are moving from one side to the another side. That is based on its permeability. Okay, means permeability of the drugs pass to biological membrane. That is lipophilic. That is in lipophilic form. Means ability of the substance is to allow another substance to pass through it. That is permeability. Okay. This permeability is indirectly based on the extent of absorption of the drug substance. Okay. It is indirectly based on the extent of absorption of the drug substance. Okay. Next, when we are considered as the drug is highly permeable, means the drug substance is considered highly permeable when the extent of absorption in human, it is determined 90% or more administered drug, that is compared to in vivo reference dose, then we will consider as highly permeable when extent of absorption is 90%, when we are considered as highly permeable. Next, now we will discuss about various permeability methods. Those are in vitro, in vivo, in situ methods. So these methods are used to determine how much amount of drug has to be absorbed, or when the drug has to be absorbed, how much amount of the drug is absorbed, and what is permeability, and also we are checking the absorption of drug. Means we can determine the absorption of drugs by using these methods: in vitro, in vivo, in situ methods. How much amount of drug is absorbed? Okay, what is permeability? So these three methods are different. So first one, in vitro method. The first method is in vitro method. What is special in vitro method? Means In vitro means outside. In vitro means outside of the body. Means here we are carried out the studies outside of the body. Okay. For conducting in vitro studies, here we are using live animal tissues. Here we are checking permeability, no? So we are using live animal tissues. Previously, we discussed about in vitro dissolution methods, no? Previous class, different in vitro dissolution methods, non-sync and sync methods are there, no? Here we are using in vitro methods. For that purpose, we are using live animal tissues. Okay. Now we are discussing about in vitro methods to determine the absorption of drug from GIT. Here we are going to use or determine permeability of the drug substances by using animal tissues. Okay, what are the different animal tissues? Here we are using the animals like rats, guinea pigs, and rabbits for the study. Why we are using animal tissues for the study? Means these are assess the major factors involved in the absorption process, and also by using these methods we can predict rate and extent of drug absorption. Means we can predict the rate and extent of drug absorption. We can predict the bioavailability. For this we are using different intestine lower experiment of animals like rats, guinea pigs, rabbits are taken for the study. Why? Because why we are taking these animals for the study means these animals are very close to the human models. Okay, the rats and guinea pigs and rabbits are resemble to the man. These are very close to the human models. That is the reason we are taking these animals for the study. Okay. Next one. Here there are different in vitro methods are there. That is physico-chemical methods. 8 methods are there. Actually, the topic is very lengthy. In your exam, you need to discuss all the things in a simple manner. We explain the figure and what is happening in each method you need to write. Okay. The first one is a partition coefficient method. In partition coefficient, partitioning of the drugs takes place. Second one is a artificial membranes. In this, we can use different artificial membranes that may be made from organic or inorganic matter. Third one is we can use the chromatographic retention indices to find out the extent of absorption and also permeability by using these chromatographic retention indices. Next, fourth one, brush border membrane vesicles. Here we can use different border membrane vesicles are used. Fifth one is isolated intestinal cells. Sixth one is a tissue techniques. In this tissue techniques, different techniques are there. Everted small intestinal sac technique, everted sac technique, circulation technique, everted intestinal ring or slice technique, diffusion cell method, cell culture techniques. Okay, these are the physico-chemical methods come under in vitro methods. First method, partition coefficient. What is partition coefficient? Means partitioning of the drug between two phases, that is oil phase and water phase. Okay, the partition coefficient is very, very important of the drug molecule. Okay, it is very important property of the drug molecule. We can determine that. So here, the partitioning of the drug between two phases, and also here we can measure the lipophilicity of the molecule. Means it has to be lipophilicity and hydrophobic. That can increase the permeability. What is permeability? Means it cross the membrane lipid bilayer and reach the bloodstream. Means lipophilicity. And already I discussed in factors affecting bioavailability. The important thing is unionized drug and lipid solubility is important. Why? Because unionized and lipid soluble drugs are better absorbed. So bioavailability is increased. Okay. So the lipophilicity is important. The lipophilicity of the drugs can pass through the biological membrane. So we predict extent of permeation. Okay. We can predict the extent of permeation. It is the measuring of the lipophilicity of the molecule. Means we can predict the extent of the drug can pass through the biological membrane. Okay. So here partitioning in this method, partitioning is takes place, partitioning of the drug between oil phase and water phase. Here we are example taken as octanol. The octanol is selected as an oil phase. Why? Because it is similar properties to the biological membrane. Okay. In this, two terms are very important. One is log P and next one is the log D. One is log P and next one is the log D. So octanol is selected as oil phase because it is similar properties to the biological membrane. In partitioning, the two terms, one is log P. Log P is an important parameter. We can predict the lipophilicity of the drug. Next, log D. What is log D? Log D means distribution coefficient. And lipophilicity tells about whether the drug is able to cross the lipid membrane. Means absorption. That indicates absorption. Okay. In partition coefficient, the ratio of how much of the drug partitioning into the water phase and how much amount of the partitioning is takes place into the octanol. Okay, that we will know. For example, if the drug partition is equally between both the phases, that is water and octanol, then P is equal to 1. Means the drug is distributed equally in both the phases, that is water and octanol. And log P is used for the measuring of the whether the drug is completely unionized. Means log P is the measurement of the partition coefficient in that condition where the total compound is unionized form. Okay, this is about log P. And log D is measured at the intestinal pH value. This will give much better idea about extent of drug permeability across the GI membrane than log P. That is about partition coefficient. Next one is the artificial membrane. The artificial membrane, it is a very useful technique for studying of passive membrane permeability. And also it is a reproducible method. It is a reproducible technique. And also it is suitable for high throughput screening. And here in artificial membrane, we are using the model name is PAMPA. Means parallel artificial membrane permeability assay. It is a method in which we can determine the permeability of the substances. We can determine the permeability of the drug substances. In this method, two compartments are there. One is a donor compartment and another one is a acceptor compartment. Here, it is a donor compartment. The above one is a acceptor compartment. Here, artificial membrane is there. Donor compartment, acceptor compartment. This is the artificial membrane. Between donor compartment and acceptor compartment, lipid is infused, that is artificial membrane. So here the artificial membrane is like a phospholipid. The artificial membrane is look like a phospholipid membrane. So what is phospholipid? Any idea? Phospholipid is a lipid that contains phosphate group. Okay, phospholipid is a lipid that contains phosphate group. It is a major component of a cell membrane. Phospholipid is a major component of a cell membrane. So here we are taking as a phospholipid membrane. And after that, the solution that is prepared by pipetting the solution of lipids in an inert organic solvent on a supporting filter. So which is placed on a 96 well microtiter plate. The solution is prepared. Next, test compound is added to the donor compartment. Test compound is added to the donor compartment that contains buffer solution. So here we are adding test component at the pH of 7.4. Okay. After that, the permeation takes place through the artificial membrane. This is the artificial membrane. The permeation takes place through this artificial membrane into acceptor compartment. So it will reach to this compartment, that is acceptor compartment through artificial membrane. Next, different samples are collected. After that, the concentration of duct permeation is measured. So different samples are collected from this uh, model for this compartment. After that, we can determine the duct permeation. We can determine the concentration of duct permeability. That is measured by different suitable assay technique. Here, for measuring the concentration of duct permeability, we can measure by using suitable assay technique. Okay, this is the figure of artificial membrane permeation assay, parallel artificial membrane permeation assay pump up. And in phospholipid consisting of also hydrophilic head and hydrophilic tail. That's the reason we are selecting here phospholipid membrane. And next, uses of this pumpa model. It is used to determine the intestinal permeability. And also by using this technique, we can predict the passive transport. And also this method acts as a transcellular permeability of the drugs. And what are the advantages for this technique? It is used for the screening of large number of compounds. And also by using this technique, we can provide information on solubility, lipophilicity, ionization status of the drug. And also it is very easy technique. Means it is which is a lesser labor intensive technique than cell culture technique. This technique is under 7 technique or 8 technique is there. All of you remember, this is a very laborious technique. That the advantages of this technique is it is a lesser labor intensive technique. These are the advantages of this technique. And next, coming to the disadvantages. So by using this technique, the approximation and oversimplification of the actual in vivo conditions are less. This is about disadvantages of this technique. Next, same technique is modification is done. That is, the technique is called as ILS, means immobilized liposome chromatography. Okay, so this immobilized liposome chromatography, many compounds with same log P have been showed to determine or to demonstrate variable membrane partitioning based on their log values. Means by using this technique, we can demonstrate the different variable membranes partitioning based on their different log values. Next, here Liposomes and lipid bilayers are produced from the mixture of lipids. And also this method is useful for the investigation of passive diffusion of the drugs. Okay, by using this technique, we can investigate the passive diffusion of the drugs through the lipid membrane. And also it is used for the passive diffusion, passive absorption of monocarboxylic acids. By using this technique, you can determine the partitionings, different membrane partitionings based on their log values, and also we can determine passive diffusion of the drugs, and also we can determine the passive absorption of the monocarboxylic acids. And another technique is again modified. That technique is called as filter IIM, filter immobilized artificial membrane. The modified version of that method is this one. And this technique is coupled with the instrument is PSR4P, permeability, solubility, retention. So by using this method, we can use for high throughput permeability screens. This method is very important. You may expect for 4 marks also. 4 marks also. Permeability method, explain in detail about artificial membranes. You need to explain Pampa method, and you need to draw this different compartments and lipid layers, everything. Advantage, disadvantage, modified techniques, coupling techniques. Next one is the chromatographic retention indices. In chromatographic retention indices, this is the one of the chromatography technique, that is immobilized artificial membrane chromatography technique. So by using this technique, along with this physicochemical parameters, it is used for the evaluation of passive intestinal absorption. Okay, these immobilized artificial membrane packings are prepared by covalently immobilizing monolayers of membrane phospholipids to silica particles. So this is the preparation of this technique. And next technique, micellar liquid chromatography. This is also used for the prediction of passive drug absorption. In this technique, the retention of drug is mainly depending on the hydrophobic and electronic and steric interactions, mainly depending on these three conditions. Why we are using different types of chromatography techniques means that chromatography techniques are easy to operate, and by using this technique, the sensitivity of the method is very high. That is the reason, in generally, for in vitro methods, for permeability methods, we are selecting chromatographic techniques. So this is about chromatographic retention indices method. Next, brush border membrane vesicles. Brush border membrane vesicles. Why the name brush border? Means here in the figure, these are the structure of microvilli. This is the structure of microvilli. So thousands of microvilli are from the structure. We can call brush border. See here, this is the brush border membrane. Brush border membrane is there. So this is formed on the apical surface of the epithelial cells. So this is formed on the, these are the intestinal epithelial cells. So this is formed on the or found on the apical surface of the epithelial cells, that is small intestine. The apical plasma membrane. So this housing, these enzymes are composed in a numerous microvilli. Where is the person? Numerous microvilli. So with extend from the cells. So this will constitute like a brush border. Okay. These enzymes are embedded in the microvilli. So this is referred as brush border enzymes. So the brush border is a name for the microvilli that will cover the surface of the epithelial cells. that is simpler columnar epithelial cells. These epithelial cells are found in small intestine. For this study, we are using both animal and human tissues. Okay. Procedure. Intestinal tissues are treated with calcium chloride, and after that we are going for precipitation method using centrifugation. After centrifugation, different types of pellets are formed. So we need to resuspend the pellets in buffer solution. So that will result in the formation of vesicles. Okay. After that, the vesicles are mixed with the drug solution. The vesicles are mixed with the drug, and that is present in the buffer solution. After mixing with the drug, we can filter the solution. After that, we can determine the amount of drug is taken up by the vesicles. Okay, that is, from that we can know the drug absorption. Okay, this is about brush border membrane vesicles. This is also important question you may expect for 4 marks. You need to draw this figure. And advantages. So this is the useful mechanic studies for drug absorption process. In brush border membrane vesicles, the microvilli that will constitute the brush border. Okay, so these are the enzymes are useful for the final part of digestion. Okay, the enzymes will allow the absorption of the digested nutrients. Okay, the brush border cells are found in the organ. What is that organ name? Tell me, what is the organ name?
Intestine.
Small intestine. So this is where absorption will takes place. This is about brush border membrane. Next one, isolated intestinal cells. In isolated intestinal cells, the small intestine is perfused with the enzyme solution. Okay. After perfusion with the enzyme solution, the cells are released. Next, these cells are treated with chelating agents or enzymes. After that, after isolation, the cells are suspended in a buffer solution. And after that, at the time of experiment, the cells are separated. These separated cells are resuspended in buffer solution. After that, we are going for shaking. At a specific period of time, the cells are separated or removed by the filtration. After filtration, we need to extract. After that, we can determine drug absorption. How much amount of drug is absorbed. This is about isolated intestinal technique. Next technique is tissue techniques. In tissue techniques, there are 4 techniques are there. Everted small intestinal sac technique, first one. In this method, isolation of small segment of intestine from the animal, that is rat, and after that inverting the intestine. After inverting the intestine and filling the sac, and sac is filled with the drug-free buffer solution. The sac is filled with the drug-free solution. And here we are isolating the segment, no? That segment having 2 sides. So both the segments are tied off. And the sac is immersed in a flask. The flask is Erlenmeyer flask. Okay. Both the segments are tied and sac is immersed in a Erlenmeyer flask. That flask contains buffer solution. Okay. In that, drug solution is there. Drug is there. And after that, after everything is set, the flask and its contents is oxygenated. Okay. And this flask, we are maintaining the temperature that is 37 degrees centigrade. Means the total setup, the total preparation is maintained, the temperature 37 degrees centigrade. And during this process, shaking is required. After that, different time intervals, the sac is removed. And the concentration of drug in the serosal fluid is determined. Or we can go for the